My name is Jason Leach. I'm a research hydrologist here at the Canadian Forest Service in the Great Lakes Forestry Center. And today, talk about a little study focused on stream temperature response and recovery in northern hardwood forests. Before I get going, I just want to highlight Danielle Hudson and Rob Fleming, particularly Danielle, who did a lot of great work on this project. Going a little faster. Okay. So forest streams provide really critical habitat for a variety of aquatic organisms, such as cold water fish. Water temperature is a key factor influencing these aquatic habitats, since it impacts biogeochemical rates, ecosystem metabolism, and fish growth and survival. And so there are concerns that climate change and forest disturbance, such as wildfire and harvesting, are impacting stream thermal regimes with consequences for aquatic ecosystems. And it's partly because of these concerns that sustainable forest management approaches in many jurisdictions specifically or specify concerns around forest activities and impacts on stream temperature. So for example, in British Columbia and Ontario, there's regulations aimed at preventing or mitigating changes to stream temperature harvest operations. And so there's been, obviously, because this is a concern, there's been quite a few decades of research on this topic. And most of this research is really from coniferous forests predominantly, particularly in pluvial regimes or in rain-dominated regimes. This is really a lot of research in this area from coastal BC, Washington State, Oregon, California, and whatnot. And the big questions that they've been tackling there is really looking at clear-cut harvest. And particularly the question around riparian buffers. Do we need them? How wide do they need to be to minimize impacts on stream temperature? In particular, and in addition, a lot of that research is really focused on short-term responses. How has stream temperature responded in 1 to 5 years post-harvest? And so we have a lot less research on deciduous forests, and in particular the sorts of harvesting approaches that are used in these forests, such as selection harvest, shelterwood approaches. In addition, we don't have a ton of research from nival regimes, that is, landscapes or forests that have a lot of seasonal snow cover. And finally, another major sort of glaring research gap is that we don't really understand how stream temperature responds to really long-term, periods post-harvest. So given these knowledge gaps, the research objective here that we're going to try to address is we're going to assess stream temperature response to and recovery from 3 different silvicultural systems. Clear-cut, selection, and shelterwood in a northern hardwood forest. So this work was conducted at the Turkey Lakes Watershed. This is a long-term research platform located just north of Sault Ste. Marie, just in from the eastern shores of Lake Superior. It's been operated since the early 1980s, and Kara Webster, who's got another talk right after here, will talk a bit more about some other aspects of the site. But in 1997, there was an experimental harvesting conducted at the site. And you can see some of the treatments here. These were done at small watershed scales. So there's an entire watershed that was clear cut, another one that was selection harvest, and then finally about 70% of the lower elevation of a third catchment was given a shelterwood treatment. In addition, there's a number of reference catchments that were retained that were not subjected to harvesting. 2 of them are shown here because these will be included in the analysis that you'll see. Now, since the early 80s, technicians have been visiting these sites, grabbing water chemistry samples, doing work on the weirs, on the data loggers and whatnot. And every time a technician visits, they stick a thermometer in the water and take a stream temperature measurement. And over time, we've got over 40 years now of these sorts of spot stream temperature measurements. And we're sort of interested, could these be leveraged to look at some of these long-term impacts of clear-cut and selection harvest on stream temperature? And now there's a number of limitations with spot stream temperature measurements. As you can imagine, stream temperature sort of varies at diurnal scales and things like that. And just a single measurement may not be that representative. I don't have time in this talk to sort of get into some of the details here, but because the measurements have been so systematic, the site visits by the technicians over the 40 years, so repeatable and systematic and whatnot, we were able to sort of deal or account for a lot of those biases in the data. A particular strength of this data set is that we have 16 years of pre-harvest data and 23 years of post-harvest data. So a really great opportunity to look at how stream temperature prior to harvest is related between these different watersheds and to account for within site or between site variability. And then 20 years of post-harvest starts to allow us to look at these long-term responses. And so I'm going to show a little bit some of the data now. I'm not going to spend too much time on this plot because I want to show you some of the statistical analysis, but I thought it'd be important to sort of show you some of what the data looks like behind the scenes kind of thing. And so what we have here is a number of scatter plots. On the x-axis is the main reference site, C32, the water temperature measured there. And the water temperature on the y-axis is the concurrent measurement made at the clear-cut selection shelterward, as well as a second reference catchment. And so the key thing here is that each point represents a measurement that was likely taken well within an hour of each other at the two sites. So that's kind of important for dealing with some of these diurnal and seasonal biases. I should also mention that this is primarily for the leaf on period, so kind of running from June to September inclusive. And so what you see here is the gray dots are the pre-harvest values, and then we've got the post-harvest colored by this legend here. And like I said, I don't want to spend too much time on here, but I do want to highlight a couple of things. One is that if you look here, you'll probably notice that some of the immediate years following harvest for the clear-cut site are generally plotting above the pre-harvest relationship. But then once you get into sort of 10 to 15 years post-harvest, those points are plotting back within the range of the pre-harvest data. In contrast, the selection harvest and the shelterward harvest, all of those sort of post-harvest measurements are seem to be falling well within the range of the pre-harvest relationships. The same is happening for the reference versus reference site, which we expect to happen, right? Although you can see that this relationship is a little bit noisier. So we're going to look at this in a little bit more detail, or from a statistical approach. And so what this plot here is showing is it's the estimated stream temperature response for 3-year moving windows at the harvested catchments, so that's the top 3, as well as a reference catchment used the methodology in that last plot. The vertical gray band here indicates the time of harvest. And for the pre-harvest periods located here, as well as the reference catchment comparison, what we're really looking for is for these estimates to straddle 0. If the estimates deviate from 0, that suggests a detectable difference in stream temperature for that 3-year period compared to the rest of the pre-harvest data. So I really want to focus on 3 key results from this. First of all, only the clear-cut site showed a stream temperature response to harvest. And although the response persisted for about 5 to 7 years, it was less than a 2-degree increase. This is pretty low compared to most other clear-cut studies, particularly those in the coniferous sites. And the muted response is likely due to these streams often drying up during late summer, as you can see in the picture there. If you don't have water, it's pretty obvious, if you don't have water in your stream, it can't be heated. And this is consistent with results from a study in Washington, where they also found that small ephemeral streams were less responsive to harvesting than larger streams. Whoa, that went too fast. Yay. Yeah. The second key result that I want to show is that the timing or recovery at the clear cut, as well as the lack of response with selection and shelterwood streams, are fairly consistent with observed forest canopy dynamics, where the selection and shelterwood harvest sites didn't see much change in effective leaf area index following harvesting, and the clear cut site returned to reference conditions within 5 to 7 years. Again, this is consistent with the stream temperature response. And finally, there appears to be a small decrease in stream temperature that occurred about 11 years following harvesting. And this corresponds with a delayed hydrologic response for these catchments, since the initial decade following harvesting was relatively dry. The harvested catchments didn't really show a strong increase in summer stream flow until about 10 to 11 years after harvesting, once we had entered a generally wetter period. So what we suspect is going on here is that this increased flow due to greater surface runoff with a recovered forest canopy may be driving this, small cooling effect. So in conclusion, what we found was that clear-cut harvesting showed a summer temperature increase that persisted for 5 to 7 years post-harvest. Shelterwood and selection harvest did not exhibit a detectable change in stream temperature. And finally, that the results that we're seeing here are consistent with forest cover and hydrologic dynamics, and particularly the fact that these are ephemeral streams and as I mentioned, that delayed harvesting response that may be driving some of the cooling that we're seeing. So sort of kind of key takeaway here is that from a sustainable forest management perspective is that these findings suggest that selection and shelterwood approaches may be effective at mitigating stream temperature response to harvesting. But I think a really critical thing is that the hydrologic context is key here. And so some open-ended questions are, would we see the same sorts of effects at streams with more permanent flow, for example? And what sort of responses would we see if we didn't have that sort of really dry period immediately following harvest? So these are still sort of open questions, and maybe something that we can talk a bit about in the discussion session, for example. Anyway, I want to thank you very much for your time, and I want to make a particularly big thank you to Wayne Johns and Jamie Broad. These are the two technicians that have been making these measurements and writing these notes in their field books for the last over 40 years. And the fact that they collected these data in such a consistent and systematic way allowed us to opportunistically use these data to answer, I think, some kind of interesting questions around sustainable forest management and implications for stream temperature. So thank you very much for your time.
Thanks, Jason and Jason. So it's great to be with everyone virtually today, and I'm also a researcher at the Great Lakes Forestry Center. And so I'm going to present some work that's come out of the long-term monitoring and the harvesting impact study from the Turkey Lakes Watershed as well. And I'm actually going to start with my acknowledgements, because really this work is not just for me or my co-authors, but really is the culmination of many years of work by past and present field and lab staff and other researchers. And for those that don't know, the Turkey Lakes Watershed is one of the longest and continuously operated watersheds in Canada. And without the dedicated efforts of many people, this important work would not have been possible. I'd also like to acknowledge that the watershed is located on the traditional territory of the Botswana First Nations. So in terms of the context to the study, there have been many efforts to investigate the impacts of forest harvesting on water quality and quantity. And we know that harvesting affects many aspects of the water cycle that then subsequently affect soil moisture and temperatures, which then influence nutrient cycling and export. But many studies have looked at short-term changes in water quality following harvesting, but the long-term impacts have really been poorly understood, particularly when there have been other environmental changes, such as changing climate and acid deposition, that could confound the impacts and recovery. And these really are critical questions to ask. Because of the importance of water quality for downstream ecosystems and downstream users, and as well water quality being a very key and important indicator of forest ecosystem functioning. So the goal of this research was to understand stream chemistry response of catchments to different harvesting regimes. And again, these are similar to what Jason described, the clear-cut, selection, and shelterwood. And we looked at the changes over 21 years post-harvest. And this is also a time frame in which climate and acidic deposition have also been changing. So the three key questions we were looking at were, one, what is the impact of that intensity of harvest on water quality? Second, what are the patterns in the solute changes post-harvest? And thirdly, how does acidification recovery and climate change confound this response to harvesting? So this is similar to Jason. This is the Turkey Lakes Watershed, and yes, it's just located north of Sault Ste. Marie for those that are not familiar with the watershed. It's produced lots of great science to inform policy ever since 1979. And the whole watershed is 10.5 kilometers squared. It's a sugar maple dominated forest, and it contains a series of 5 interconnected lakes. And there are 13 first order catchments that are monitored. So since this is the watershed study, a mass balance approach is used, which means that inputs from precipitation and outputs from streams are measured. So the precipitation and its chemistry is measured at the Air and Precipitation Monitoring or CAPMON site, as well as a MET station that is located just outside the watershed. And the streamflow and chemistry is measured at V-notch weirs at each of the catchments. Stream flow is monitored continuously, and we get daily to bi-weekly grab samples of the stream water that are analyzed at the water chemistry lab at the Great Lakes Forestry Center. So what changes have we observed in the environment over the 43 years? Well, temperatures, which is shown on the top panel there, have increased primarily in the late summer and fall. Changes in precipitation, which are the bottom graph, are not as well pronounced. There have been cycles of wetter and drier periods over the record. So in terms of the deposition of acidic pollutants, here's actually a good news story. These acidic deposition has actually decreased over the record. And so the top panel is shows the decrease in nitrate over the period, and you can see that it really only started declining in the early 2000s, but sulfate, which is shown on the bottom, has actually consistently declined since the beginning of the record, and this is the result of implementation of the Clean Air Act in the 1990s. So a little bit of a good news story there, that sometimes implementing policies do have good effects. So so these changes in environment have affected the stream chemistry in the catchments. And because we have such a nice long record, we can look at how those changes, what's happened with the changes in the stream chemistry. And this is really the focus of a 2021 paper in hydrological processes that I've noted at the bottom there. But I'm going to just highlight a couple of the key findings from that paper that are relevant to this presentation. So first of all, there was quite a bit of variability among the solutes. Some solutes decreased, some increased, and some were just variable. And the second point was that responses were not always linear over time. And the main take-home message from that paper was that yes, acidification recovery is occurring, but it is not complete. And it was often difficult to separate out climate effects and acidification recovery effects. So that is kind of the environmental backdrop to the Turkey Lakes harvesting impact study that Jason has already talked a little bit about. As he said, the harvest was done in 1997, and there were the 3 treatments that were applied: a diameter limit clear cut of trees greater than 20 centimeters diameter, which is not typically an accepted practice in this type of forest, but was mostly as a sort of an experimental treatment. There was also the single tree selection harvest, which is an uneven age system where mature and undesirable trees were removed. And then there was the even-aged uniform shelterwood system, where about 50% of the mature trees were harvested, with the residual trees remaining to provide shelter. And there was also some unharvested control catchments. So the streamflow response to harvesting was previously reported by Jim Buttle et al. In 2018. And the key take-home message from that work was that the runoff did increase post-harvest and that it has not completely recovered. And in fact, this is work that Jason Leach continues to look at it as part of his research. So now what I'm going to show you is some of the key patterns of the harvest impacts on the stream chemistry. And so the next few panels are set up in a very similar way. So I'm just going to explain the graph layout a bit here. So you can see the solute name is at the top. The treatment catchment is listed below that. And then the y-axis is the regression coefficient. So that's the harvest effect on the solute concentration relative to the pre-harvest relationship. And then the x-axis is the year since the 1997 harvest. The bottom panel shows control versus control comparison post-harvest, but I'm really not going to have time to discuss that. And so this work was just actually recently published. So if hopefully if you're sufficiently intrigued, I encourage you to look up the publication, as I won't have a lot of time to get into some of the details. So there were really 5 main response patterns in the stream chemistry. The first pattern was this elevation or depression of the solute concentration relative to pre-harvest, and that then returned to the pre-harvest levels. So this was seen for many of the cations, and this is likely driven by the low nutrient demand due to the tree removal initially post-harvest, but also because of increased solute production from easily decomposable harvest residues, as well as high mobility and hydrologic conductivity with the post-harvest wet up. And as you can see, the solute concentration recovers fairly quickly as the vegetation regrows. The second pattern is the elevation of the solute concentrations followed by a decline, but not completely recovered. And this was seen for potassium. And similar to that first pattern, this is driven by sort of lower demand for that nutrient initially because of the removal of vegetation, high solubility, mobility, and connectivity, but as well as input for mineral weathering. The third key pattern is the elevation of the solute concentration, but then followed by a subsequent depression in the concentration compared to the pre-harvest level. And this was seen for nitrate. And this is similar to the other solutes in terms of that initial response, but the concentrations fall below pre-harvest levels due to the high demand for nitrogen in early succession. The fourth pattern is this sustained elevated concentration, and this was seen, for example, with dissolved organic carbon. And this response, sort of in contrast to the other preceding patterns, suggests that the nutrient demand during revegetation is less of a driving factor, and that it's really the solute production from the decomposition that is more important, as well as the fact that DOC can be quite mobile during that post-harvest wet period. The last pattern that I'm just going to show you here was really one that there was sort of indifferent or a noisy response. And this was seen for total phosphorus as example. And this was a result of low or no demand and low solubility and or mobility. So to summarize the key findings here, the first is that the impact of the harvest treatments on the solutes is proportional to the intensity of the harvest. So the clear cut had much larger impacts than the partial cutting methods of either selection or shelter would. However, the selection appeared to have a slightly larger effect than the shelter would, and this is a bit counterintuitive, but there were some several confounding factors that may have explained this. And I'll direct you to some of the discussion in the paper if you want to learn more about that. The second key finding of this is that the impact and the recovery of the solutes really is a balance between these solute-specific changes, not only in nutrient demand, but sources and hydrologic conductivity. So we have to consider all the different ways that nutrients are mobilized and move throughout the soil. And so some solutes had a greater demand by trees, while others were more soluble or mobile. And through this, we identified sort of 5 distinct patterns in those changes in recovery. The third kind of interesting things is the idea of sort of these cumulative effects of different stressors. So we could see that the shorter term harvesting impacts could either offset global change effect for some solutes, while others, it augmented the global change effect. So for example, for cations, the harvesting increases initially their concentrations, while global changes from acidification and potentially climate impacts resulted a decrease in solute concentrations. Whereas this was opposite for dissolved organic carbon, where both harvesting and global changes both cause increases to those concentrations. So my last slide here, in summary, we do see resilience in terms of the water quality response to low levels of disturbance. However, it's really important to point out that the impacts of harvest on stream chemistry is confounded by changing climate and deposition. And it's certainly clear that what comes out of this is that there are still crucial questions that remain about the persistence of some of these effects over the long term, and how they may accumulate in downstream aquatic ecosystems across regional scales. So thank you very much, and feel free to contact me if you have any other follow-up questions. Thank you.
Hi, everyone. Yeah, and thank you for this opportunity to share with you some of our results from the study that was aiming at understanding the impacts of forest management on stream ecosystem integrity, and specifically on how these effects accumulate as we move downstream. As you can see, many co-authors were behind this work. Okay, so the effects of forest management on headwater streams are quite well documented, like several studies for many years have been conducted. One of those studies was the one we conducted in northern New Brunswick, where we looked at several indicators and how they changed as forest management intensity increased. And we saw an increase in terrestrial materials that were delivered to the streams, such as cations, carbon, sediments, and this had biological effects, such as a reduction in leaf decomposition or increase in biofilm biomass. But a question that arose from here, and something that is not as well studied in the literature, is what happens with these effects as we move downstream? Do they accumulate or do they dissipate? So to answer this question, we conducted this study in northern New Brunswick, and we selected 3 basins with different types of forest management. Forest management. So we call them intensive, extensive, and minimal. Intensive meaning that there were more interventions, silvicultural interventions, such as planting, soil preparation, and so on. And with this selection, we wanted to see whether stream condition changed among these different forest management types. And then within each basin, we selected 6 sites that increased in catchment size. And with this, we wanted to see how the stream indicators change from upstream to downstream. And then combining this information, we tried to see whether we could see cumulative effects or dissipative effects. And how did we do this? So we looked at how an indicator changed from upstream to downstream in a basin, and then compared the reference basin to the disturbed basins. So if we saw, for example, that the differences between the disturbed and the reference basin increased as we moved downstream, then we concluded that that effects were accumulating longitudinally. On the other hand, if we saw that differences between the disturbed and reference basin decreased from upstream to downstream, then we concluded that the effects were dissipating. And to characterize stream ecosystem quality, we used a multi-indicator approach. So we looked at water temperature, water chemistry, then mercury, which is a pollutant of concern in aquatic systems, sediments, leaf litter breakdown, an important function of streams, algae and biofilm growth, benthic macroinvertebrate communities, which are very good indicators of the quality of stream ecosystems, then food web dynamics, who is eating what, based on stable isotope analysis, and finally sculping condition. We chose sculping as fish because they are very good sentinel species for environmental monitoring. Okay, let me show you some results. I will start first by looking at the differences among the 3 management types or the 3 basins. So as expected, total disturbance was greatest in the intensive basin, and by total disturbance we mean partial harvest plus clear cut plus regeneration practices. And the number of stream crossings was also greatest in the intensive basin. However, the extensive basin in light blue was the basin with the highest percent clear cut, as well as road density. And maybe these differences explain the differences we saw in some of the abiotic indicators. So here the first panel shows inorganic sediments. And as you can see, the extensive basin had the greatest sediments, as well as dissolved organic carbon. And because these materials facilitate the transport of mercury to streams, it is not surprising that then in the panels below, the first one, that the concentration of mercury in water was highest in the extensive basin. And these greater mercury concentrations made it into the food web, because as you can see in the second panel, in biofilm, also the concentrations were highest in the extensive basin, so in the base of the food web. However, as we moved up in the food web, mercury concentrations, the increase in mercury concentrations throughout the food web were lowest at the extensive basin. So the differences among the three basins became smaller as we went up in the food web, and we did not detect differences among the basins in fish. And there were also some biological differences. So the stream function of leaf litter decomposition on the first panel was highest at the extensive basin, followed by the intensive, and lowest at the minimal basin. This could be related to the greater number of aquatic insects that we found in the extensive and intensive basins, because alongside with microbes, these aquatic insects mediate the decomposition of leaves in streams. And at the same time, we saw that, well, probably the greater number of insects also explains why sculpin in the intensive and extensive basin had more energy for storage and reproduction. So here we are showing sculpin GSI, gonadosomatic index, which indicates how much energy the fish has for reproduction. However, the minimal, in terms of biodiversity, the greatest number of aquatic insect genera were found in the minimal basin, then the extensive, and the lowest richness values were found in the intensive basin. Let's look now at the downstream effects. So we saw both accumulation and the dissipation of effects, depending on which indicator we were looking at. So for inorganic sediments here on the first graph, so on the x-axis we have from up, the sites have been sorted from upstream to downstream. And as you can see in light blue, the extensive basin, there is an increase in inorganic sediments as we move to larger downstream systems. The two lines represent two different sampling years. And the increase, the downstream increase in the minimal and intensive basin were not as pronounced. So that means that differences between the extensive and the other two basins became greater as we moved to downstream sites. In terms of the diet of benthic macroinvertebrates, we also saw some indication of accumulation of effects. So the green line represents what we saw in the minimal basin in terms of the proportion of the diet based on algae of the aquatic insects. And this increase that we saw in the minimal basin is what you would expect in a natural system, because as you can imagine, as the streams become wider, more light gets in, so more algae grow, and then there is this shift in diet. However, in the extensive and intensive basins, for some taxa, we didn't see this transition towards a more algae-based diet, or a weaker one. So therefore, we concluded that forest management seems to alter the diet of stream consumers and makes these diets more terrestrial. And this has implications for animal growth and reproduction, because algae is considered to be of better nutritional quality than terrestrial food sources such as leaves. But we also saw some dissipation of effects. For example, for water temperature, in green, in the minimal basin, we saw quite a pronounced increase in temperature as we move downstream, which also makes sense if the streams get wider and more sun gets in. But this increase in the other basins was either weaker or non-significant. So this means that the differences in water temperature detected in the small streams disappeared as we moved down the river, or were even inverted. So the low temperatures in the minimal basin that we saw in the headwaters became that basin had the warmest water in the larger downstream rivers. And finally, for mercury concentrations in some aquatic animals and food sources, we saw an increase in the concentrations as we move downstream, but only in the minimal and intensive basins, not in the extensive. You can see in these two plots how in blue light, the extensive basin, there was a decrease in mercury concentrations. FPOM, so this is a fine particulate organic matter, a food source. And then this is some filter feeder hydrocyclics. This means that the differences observed in the small streams, with the extensive basin having the greatest concentrations of mercury, then disappeared as we move downstream. So there was a dissipation of the effects. So in conclusion, we can say that forest management effects are not limited to small streams with an intimate connection to the surrounding land, but also extend downstream. We saw that some of those effects accumulate downstream, whereas others dissipate. Interestingly, we saw that the impacts of forest management on streams were generally greater in the extensive basin than in the intensive one. So this means that greater overall intensity of forest management does not necessarily mean greater environmental impacts, especially when considering clear-cut versus partial harvest, because the extensive basin had more clear-cut than the intensive one. So that could explain many of the results we are seeing. And the last main conclusion is that while best management practices in northern New Brunswick do not eliminate all effects of forest management, we detected some effects, they do still seem to protect biological integrity downstream, because all sites that we studied were classified as having good or very good biological quality based on macroinvertebrate communities. So thank you, and yeah, happy to answer any question you may have.
Thank you, Jason. Thank you very much for the presentation. Thank you, everybody, to be here in this room. First of all, I want to tell that these results of this work are the result of one master thesis of one of the former students, Armando Falfán Cortés, and he actually is very busy in his new job, and he was not able to attend this important meeting. So he asked me to present the result of his research, and we also co-authored this with Valentin Reyes and Aurelio Fierros. Both of them are presented in this time. Natural regeneration of forest stands under management is one of the most important and relevant silvicultural practice. To be successful, this requires extensive knowledge of the factors involved in the regeneration and also making proper site preparation. In Mexico, 50% of the forests under management are treated with uneven age methods, basically with the selection method. And the other 47% are treated with even age methods, basically with the seed tree method, silvicultural system. This method in particular was started in the end of the decade of the 1970s. Actually, I remember the Mexican government started to apply this method in 1977, because before this year, all the forests in Mexico were harvested under the selection method, only the selection method. And recently, in the last 10 years, in some places, people also apply the clear-cut method. For example, here we can see here this picture in Puebla State. And in some places, like in Puebla and Jalisco State, they also leave some variable rotation trees to protect some of the ecosystem values in this forest. But back into the seed tree method, we have very successful examples in Mexico of the application of this method. For example, in these two slides, I'm showing two sites in the state of Hidalgo, in the central part of Mexico. These stands were the result of the application of the seed tree method. You can see very, very nice density of the trees. And the only thing that I remember they did a very, very good soil preparation. Despite of this, in many places, people plant seedlings under the seed trees. Even though this activity is not included in the rules of forest management in Mexico, because the use of forest plantation for a new stand is only considered when natural regeneration is not achieved after 3 years. So this was the reason, because Armando, my former students, did this research work, because he noticed when he former job, he had to go to the field, to make a survey to evaluate the condition of the seedlings, and he noticed that the natural regeneration, because he found a lot of natural regeneration under the seed trees, and also he was evaluating the planted seedlings. So he found that natural regenerations usually have better performance than planted seedlings. So he did this work to compare growth characteristics between natural regenerated seedlings and planted seedlings in 3 cutting years. And also he evaluated the water potential in both types of seedlings. This study was done in the Ejido Carbonero Jacales, in the municipality of the Aguayacocotla, in the Veracruz state. He is the Veracruz state. And this Ejido is very close, is neighbor of Puebla state. And this is part of the strip called the Cloud Forest, coming from the north of the state of Veracruz, Tamaulipas, coming through the south until Oaxaca state. So this is very, very special kind of forest in Mexico, very important, because there are many species that are used to industrial, of the wood industry. He located 3 areas of cutting areas, one in 2014, one in 2015, and one in 2016. We can see here, this is the area of the Ejido, and we can see here the 3 areas that he found to make the study. The study area has these characteristics in topography, in climate, and soils. And we can see here the ecosystem basically is dominated by pine and pine oak fir forest. And there's a lot of wildlife, some species that we can find in this area: squirrels, rabbits, gophers, possums, reptiles, and birds of prey. So as I said before, this characteristic is very common in this kind of cloud forest. The 3 areas that he chose, we can see here that has almost the same physiographic characteristics. The 3 areas that he located, the exposure was the same, the altitude was almost the same, and the slope also was similar. So he measured the annual growth of natural regenerated and planted seedlings, and this was compared in a paired way to target seedlings in these areas. So the age that he used the age of the seedlings was 4 years old on average. How was calculated the age? Well, for natural regenerated seedlings, he made a destructive analysis and he counted the annual rains. And for planted seedlings, he took the age, the year when the seedlings were planted. So he made 4 sites in each area of 100 square meters, and he made the study in 2 field seasons, from June to July in 2018, and from May to June in 2019. He had to identify the origin of the seedlings. The natural regenerated seedlings, the way that he decided to choose is because they were random distributed. And of course, the planted seedlings were distributed in homogeneous condition, keeping the distance of the origin of the plantation and over the same line. Obviously, he tried to find a pair of seedlings, both of them, in order to make the comparisons. In this way, he took the total height in 2018 and the total height in 2019, and the diameter in the stem base for both years. At the end of the dry seasons, that means in May of 2019, he evaluated the water potential of the seedlings. For this, he took 6 seedlings, 14 per site, sorry. That means that he has 18 seedlings to measure. In these seedlings, he measured the water potential with the pressure chamber on the twigs, of course, in 3 times. One, before the sunrise, from the 4 to 6:30 in the morning, and the second one at the noon time until the 2:30, and the time 3 was in the sunset from 5:30 to 8 o'clock. He took 3 days to do it, one day for each year of the measure, 2014, 15, and 16. And then he took the water potential. He also measured only one time, unfortunately, the moisture content of the sites. For this, he used 12 sites, and he took the moisture content at the 10 centimeters depth. The statistical models that he used was for the diameter and for the height. It was the generous linear mixed model to see the difference, and also he applied the least significance difference in order to see the difference between treatments. And for the water potential, he used the mixed effects model with repeated measures, and also he made the least significant difference in order to see the difference between the treatments. The basic results, we can see here the analysis, the statistical analysis. He found significant difference, statistical difference in the 2 variables, in height and in diameter, considering the origin and the age in both cases. And in the case for the water potential, he only found a significant difference in the variable time of the day, but not for the origin of the seedling. And he also found the interaction origin time that was a significant difference. So if we see here the graphs, we can see here the greater annual height growth for natural regenerated seedlings, and compare with the planted seedlings, there is a difference. And the variation between 2 kind of seedlings was lower in the natural regenerated seedlings. Same pattern. We can see the same pattern here for the diameter. The same consideration. And in the case of the water potential, this graph, we can see here that at this time of the day, the seedlings had a better hydration condition, but at no time they had a very, very high water potential. The plants are very stressed, but there is no difference between both seedlings. At the time when the day is advancing and the temperature is lower, we can see here that the natural regenerating seedlings had better condition to recover from the stress. Obviously, this probably needs more measurements to the long of the growing season, because as we see in this graph, this higher water potential may be explained by the result and the soil moisture is lower in this time of the year. And here start the growing season, and you can see the precipitation is increasing. And probably we need to do more measures at this time. But you know, students are limited by time. He had to finish his field job, his field work, in order to write the thesis and be graduate, because there are time limiting to do that. Otherwise, probably he couldn't get his master's degree. So what are the general conclusions? The general conclusions are really easy, as you see. Seedlings from natural regeneration show better performance. That means they have greater height and diameter growth. And also the seedlings coming from natural regeneration have a better physiological performance to recover from water deficit. But I think that the most important issue, or most important conclusion of this job is that we can recommend to the producers that they don't have to plant if they are using the seed tree methods. What they need to do in that forest is make a good soil preparation, and also try to schedule the final cut with the dispersion of the seed season. They will save a lot of money because they don't have to produce plant in the north, but they still continue to do it. In the case of clear cuts, that is justified because they have to plant immediately after the final cut. But in the case of the seed trees, I think they don't have to plant. I found a lot of information in the literature that they report similar results like our results. Finally, these results were published in this journal, Revista Chapingo, Serie de Ciencias Forestales y del Ambiente. This is in English and in Spanish. It's bilingual journal. You can find these results in Google, for example. And then, pues the last thing that I have to say is gracias, thank you, and merci beaucoup. I don't know how to say in French, only merci. Thank you very much for your attention.
First, I'd just like to acknowledge the research team involved in this project. This project did wrap up in 2018. There was a master's student named Adam Matheson that worked during the second year of the study, and Derek McKenzie was the university thesis supervisor under that project. So they've been engaged with this through parts of this work, and hopefully we'll make some sense of things and explain what this is all about. So motivations to hitchhike. There's lots of motivations to hitchhike. So I'll go through a couple of them here just to be brief. So we do know that nursery stock planting is a reasonably reliable way to get plants, and this has historically been trees, out onto disturbed sites, whether these are harvest blocks, industrial disturbances, this type of thing. We haven't applied this concept as widely to native herbaceous species, but it is being done, at least from what I know here in Alberta. It's being done in reclaimed industrial disturbances a little more frequently. We had this idea that there could be the potential to look at combining trees with native forbs for a 2-for-1 planting idea. So this would make it a little more cost effective, and there may have been some additional opportunities for synergies. And the other benefit would be a quick and dirty increase in plant diversity. So if we can plant 2-for-1 seedlings out onto reclaimed industrial disturbances, we double the diversity right off the bat. And what we see here in this image is just an example of disturbances we tend to see throughout northern Alberta at final reclamation. We get a lot of flux of non-native agronomics, lots of graminoids, and we don't always get a big flush of native forbs. So hitchhiking is a way that we could look at getting more of this native forb coverage early on. And this may have downstream effects by supporting the facilitation and ingress of other species, because you're going to attract a different biota by having more of these flowering plants across this landscape. So this concept really, I think, applies best potentially to industrial disturbances. We don't tend to have the same vegetation management challenges in the same way in post-harvesting sites. So I'm sure there's some exceptions. So I've just listed a few examples here, but this is not an exhaustive list. And that really brings us to the purpose of this study. So before we can recommend and tell folks to go hitchhiking their trees and forbs, we need to figure out how to grow these things together. So there isn't tons of data on growing native forbs as nursery stock anyways, and then certainly not in combining them into a common plug. So for most ecologists and plant physiologists, the idea of putting these species in close competition is at first glance perhaps a no-no, but I was optimistic that there could be some potential ways to do this. So this study is really looking at manipulation of stock characteristics in order to successfully grow these things. And at the end of this presentation, we really want to try to ask 3 questions. So the first is it possible to produce these mixed species or hitchhiker container stock types so that they both result in growth and persistence in the field? And then the second question that's related to the first is how critical are those stock production considerations? So how critical is cavity size? How critical is that timing of sowing in terms of how you introduce that species in to the mixed species stock? And then the third question is, are there other benefits? And are there other benefits in terms of mitigating vegetation dynamics around our target tree species? So can that native forb reduce the development of other graminoids or other non-native herbaceous species that tend to come into these industrial sites? A little bit about the nursery stock production. So we have in this trial 1 woody plant. That's the target here, and this is white spruce. And it was really chosen because of its known tolerance for a wide range of light conditions. And we expected that it would better tolerate the growth of a companion herbaceous species. The choice of herbaceous species that the woody plant would be grown with was based on what we knew from commonly occurring forbs in the region and ones that we found in post-industrial disturbance. So we see these in ditches, we see these in disturbed landscapes. So both showy aster and fireweed are common in the region, but they exhibit different growth forms and different morphologies. And these seedlings were grown in our research greenhouse in Peace River from March to November 2014. And then we lifted them and put them into overwinter cold storage, and we planted them in May 2015. And this trial, again, is looking at one of 2 different native forbs being mixed in with the woody plant. And then we have 2 cavity sizes, a 220 mil and 340 mil cavity volume. And then we also have 4 sow dates as a cross factor. So we sow the forb in 6, 8, 10, or 12 weeks after sowing the white spruce. So this is going to create different levels of potential competition between the white spruce and the forb. And lastly, although we're not comparing these things directly, we did also singly grow white spruce seedlings in a 125 mil cavity. This is a more conventional cavity for that's often utilized in these types of sites to plant white spruce. And they were produced as a secondary reference to understand something about the site conditions and understand relative growth, though the intent was not to compare these directly. They are not the same. So we can talk a little bit more about the philosophy of these comparisons later, but that's what was done in this trial. There was 4 study sites that we planted these seedlings into, and they were 50 to 60 kilometers northeast of Peace River. All were reclaimed in 2013-14 and then planted in spring 2015. 21 seedlings of each stock type combination were planted at each of these sites, and this leads you to some hierarchical data. So we have them planted in lines and numbers within lines. So that is that. On to field measurements. So this is brief and providing lots of information, but I really want to try to give us some time to talk about the results. So the important thing to know about field measurements and to consider as you examine and interpret the data set is the cover data is based on a 1/2 meter by 1/2 meter quadrat. I show an example here on the right, and you can see the white spruce seedlings. So the blue arrow is always going to point to white spruce. The red arrow is to the native forb. In this case, it's the showy aster. And so we're assessing cover in this quadrat centered on the white spruce seedling. So if we have aster creeping out or fireweed creeping out, we're actually underestimating its coverage. In terms of data analysis was complicated. But I'm not going to go into the details. We are using our statistical software for this project. We used a variety of linear and nonlinear mixed effects models, and that was really to apply an appropriate distribution to these data sets. And I make notes of this as we go along in the notes section. We also do analyze the single grown white spruce seedlings so that we can not directly compare means, but we have similarly statistically treated data when we're making reference to it. So these are just with the control data, it's just a random effects model. And I present this with the white spruce characteristics as we go along. So to our first question, is it possible to produce a mixed species container stock that results in the growth and persistence of both species? So we'll look at this from the viewpoint first of survival. And what we see in the table on the right here is that both fireweed, so white spruce that's grown either with fireweed or showy aster, have very similar cumulative survival outcomes. We're at about 80% or 0.8. Cavity size, also a similar effect, and we're around 80% again. There was no significant interactions amongst these treatments. But what is important is that sow date. So when we sow the forb in very early, so we're getting a large forb and a very small white spruce seedling, we end up getting a little bit of a decline in cumulative survival relative to the other sow dates. And so here we're around that 80 to 85%. And then our single grown reference spruce again is having a little bit higher average at 0.9, but a larger range in terms of its confidence interval. And that is really due to strong site differences. So when we look at the probability of occurrence around the white spruce, and I say the forb probability of occurrence rather than survival, because I think there is some uncertainty in this data. We're using a quadrat-based assessment on the white spruce seedling. If that forb is potentially still present, but it's migrated away from the original planting location, which is possible, we may be underestimating survival. So we call it occurrence because I think it's more appropriate in this particular condition. And for this, we can see here some clear trends. So for showy aster, higher probability of occurrence, higher survival with early sow dates and larger cavity sizes. But with fireweed, no real difference. So neither sow date nor cavity size was very important. We move on to the second question. How critical are those stock production considerations, cavity size, timing of sowing, when we develop this container stock and watch it grow in the field? And for this, we'll look at some of the growth data. So if we look at cumulative height increment, that's the top series of panels, and then above ground biomass in white spruce. So this is all pertaining to white spruce. We see a couple of clear trends. So first, we get an increase in height increment, cumulative height increment with later sow dates. That's again, you're going to conversely have a smaller forb as the sow date becomes later. We don't get an effect with cavity size, and we get a modest effect with fireweed versus aster. So the cumulative increment's a bit higher with spruce grown with fireweed. And the above ground dry mass follows a very similar trend, though there are some slight differences. So it's more of a break in dry mass between early sow dates and late sow dates. And we do get a small cavity size effect and again, a small forb effect. If we look at coverage of the forb, so coverage in the quadrats, what we see here again with showy aster along the top panels is a opposite trend to the white spruce, where you get that decline in cover as you progress into later sow dates. Again, the showy aster is very responsive to cavity size. And then when we look at coverage over time, because I think this is important, is persistence, we see that coverage stabilizes by about the second year of the study in showy aster and doesn't really change too much thereafter. In fireweed, we don't get a lot of change in coverage with sow date, but there's a little blip around 8 weeks, but fairly similar overall. Again, no change with cavity size and no effective change over time. So that coverage around the white spruce was pretty consistent and pretty constant across the 4-year study. So this brings us to our last question in our little whirlwind 15-minute presentation. And really, our last question here asks, do we see differences in vegetation coverage surrounding that white spruce when we hitchhike seedlings? And for this, we will actually compare that to our singly grown white spruce seedlings that had no hitchhikers. And for this comparison, we're going to look only at one stock type. So we're looking only at the stock type with a 10-week sow date and 340 mil cavity. So that we don't have unbalanced data in terms of having too much data on the hitchhiker and not enough on the control. And we see some potentially non-surprising, but a little bit surprising results. So first with native forbs, this is not surprising. We hitchhike these plants and we get higher native forb coverage. And this is clear in panel A. And is not terribly surprising. We do see a little bit of benefit with the aster. The aster coverage is driving a bit higher total native forb coverage. In grasses, we see some interesting trends that are mixed in with time. Early on, our control seedlings, so no hitchhiker, have lower grass coverage surrounding them, but as time progresses, this coverage seems to be increasing. Conversely, the hitchhikers have a little bit higher grass coverage, but it is stabilizing by around the 2nd year and possibly declining at year 4. In terms of non-native forbs, there's a general decline over time, but converse to what we thought we would find, a little bit of an increase, though it's quite subtle with the hitchhike stock type. So we get a little bit more non-native forbs relative to our controlled treatment, and this might be partly driven by that increasing grass dominance in the controlled treatment with time. So in summary, looks like I got 1 minute here. To our first question, is it possible to produce that mixed species container stock? I think the answer is yes, we can grow these species together. We can get reasonable survival or occurrence outcomes up to 4 years post-planting. And how critical are those stock production considerations? They're quite critical. So timing of sowing in this trial was really the most important variable. Cavity size had some importance in some situations, but timing of sowing was the stronger factor. And do we see evidence that hitchhiking is mitigating the development of some of these undesirable herbaceous species around the white spruce? No, we don't see that in this particular data set in this time frame. So it doesn't mean that it's not having an effect, but in the way we measured it and over the time frame we looked at, we don't see evidence for it. So with that, I want to acknowledge funders and staff and students for this work and for all of their hard efforts in this 4-year project, and a few other citations that I couldn't fit in the notes. And with that, I can answer questions or move on to our next slide.
So for the final presentation, I'll be looking at seedling launch box concept. So we're looking at a circular economy solution for incorporating organic amendments into nursery stock seedlings. We have some project teammates, including Stefan, Arangsa, Paulo Muson, and Amanda that presented initially. So the question is, why do we give seedlings a launch box? It is clear that during resource exploration or extraction, forested lands are disturbed. So one of the key factors to achieve successful reforestation is to have nutritional topsoil or nutritional quality topsoil. For us to have this, mostly you get to broadcast inorganic fertilizers to encourage growth of native species, but you find out that you also encourage the growth of highly competitive non-native species or the weeds. And when you get this, there's a potential of slowing down the natural succession of native species. So what, if we have this, what can we propose that can be able to move away from this method and get us to have even higher quality seedling stocks overall. So we're looking at having a nutrient loading concept. So the launch box, what we term that launch box, is kind of giving the seedlings, the new seedlings, a little bit of launch packs that we can take to the field to them. The initial concept was to have quality seedlings to be able to take them to the field and for them to outgrow non-native species and for us to get to our restoration purposes. But because of, while looking at it, we figured out, okay, we can incorporate the circular economic aspect. So how can we do this? So much as less we can have our organic amendments and we can incorporate them in our nursery stock production. It can also be beneficial for diverting organic waste material from landfill as much as supporting a circular economy. So generally, the aim of this project is to develop a solution rooted in circular economic principles to facilitate improved establishment on reclamation on reclaimed industrial sites using organic amendment. To achieve this aim, we'll have two specific objectives. One of them was to determine whether alfalfa pellets or hydrochar can be incorporated into organic substrates during nursery stock production. And secondly, to determine if the additional launch box of alfalfa pellets or hydrochar that we provided during the nursery stock seedling production can improve growth and survivorship in a reclaimed soil. A quick overview of the two amendments, organic amendment sources we use. We use the alfalfa pellets and the hydrochar. Alfalfa pellets, they are plant-based pellets that were produced from the alfalfa plant. And this was sourced from alfalfa grain by Western Alfalfa Milling Company in Saskatchewan. The second organic amendment we used was the hydrochar, which was produced by hydrothermal carbonization of a biomass in a pressure reactor at about 200 degrees centigrade. This was produced by one of our research teams at NAFEW at Edmonton. These two organic amendments have two distinct properties, most especially how they were prepared. The alfalfa pellets were prepared at 600 degrees centigrade. I will have a hydrochar prepared at 200 degrees centigrade. While we use lower temperature for the hydrochar was to make sure that all the elemental composition of the feedstock or the biomass we use for the preparation remained intact, that we didn't lose any nutrient that ordinarily we will have from the original biomass. Next to the method, we use 5 application rates for the alfalfa pellets and 4 rates for the hydrochar. For the alfalfa pellets, we use 0, 1, 5, 10, and 20% of the cavity volume of our our styroblocks. And the hydrochar we use for concentrations, 0, which is the control 1, 5, and 10% of the cavity volume. We tested 2 plant species, popular streamloids, otherwise known as aspen, and our Betula papyrifera, otherwise known as pepper birch. We tested these 2 species because they are widely distributed and they are largely planted in reclamation sites. We looked at reclamation soil, and the reclamation soil we chose was a high quality reclamation soil. When we call them high quality, it's actually a reclamation soil where land preparation has been completed for revegetation studies. So they have high nutrient and high organic matter content. And just a quick one for this, for the preparation of the amendment, we'll fill them up to 80% cavity volume with the nutrient, with the hydrochar or alfalfa pellets, and the remaining 20% will cap it with peat. We have 2 experimental phases, just like our objective was. The phase 1, we looked at seedling production and 32 replicates of each of the treatment, that is aspen or pepper birch. We are grown in 412 styroblocks planting containers, and 16 plants were destructively harvested to look at the physiological properties, and the remaining 16 replicates were transferred to the phase 2 of this study in a reclamation soil, also in the greenhouse. Just a quick photo of the phase 2 experiment. I think these photos were taken during harvest. On the right-hand side is our pepper birch, and on the left-hand side we have our aspen growing in reclamation soil in greenhouse, in regular pots in greenhouse. Quick overview of our phase 1 results. So we looked at seedling height and root collar diameter. It is clear that the incorporation of alfalfa pellets at higher concentration, up to 10%, increase the seedling height as well as root collar diameter at the seedling stage. Also, we found out that hydrochar did not have any effect on the seedling height or root collar diameter overall. Also, we looked at the root and leaf mass of aspen and peppergrass in the phase 1. So we found out that up to 5% concentration of alfalfa pellets increased the leaf and root mass of aspen or peppergrass. So just like our seedling height and root collar diameter, the hydrochar did not have any effect on the leaf biomass. After the phase 1, we got to the phase 2. The phase 2 incorporated those phase 1 trials that were planted in reclamation soil in the greenhouse. After 6 months of trial, we looked at different properties of different zoological properties of these plants, both aspen and peppergrass. And from what we see on the slide, the leaf biomass and the stem biomass of aspen, including the shoot height and root collar diameter, including the shoot height and the root mass, we observed similar patterns just like we had in the phase 1, but with a little bit distinct differences. So the alfalfa or hydrochar aspen seedling, they did not outperform even the regular seedlings. But we can notice with the stem mass that the stem mass was significantly increased with alfalfa pellet. Just like we had with the phase 1, we did not see any differences with our hydrochar. Significantly, we looked at 20% alfalfa loaded seedlings, you could see that there were significant differences when you compare them to the regular or standard seedlings, but we also did not find any differences in root biomass with our hydrochar. Back to our peppergrass for the outplanting stage, so we looked at similar properties and we'll find almost the same differences as we see in our aspen, with alfalfa pellets or increasing increasing the stem biomass, alfalfa pellets increasing the root mass, with no significant difference observed with hydrochar. We also looked at the root mass at up to 20%. You can see that we found differences with alfalfa pellets. However, with our hydrochar, the results of the root mass were similar to what you will have with our standard seedlings. So with this quick result, we could conclude that the growth response to nutrient loading parallel the rate of loaded amendments, especially when you look at alfalfa pellets. So specifically, we know that application of higher loading rate of alfalfa pellet, most especially when you get up to 20%. And when I say 20%, is at the maximum rate that we tested. They show superior stem and root growth when you compare them to standard seedling during seedling production and even during outplanting. So for the two phases, we found differences at 20% alfalfa pellet application rate. However, our hydrochar amended seedlings, they showed no difference in growth compared to standard seedlings, whether it was during the stock production or during outplanting. But we understand or we're figuring out that these could be related to the properties of the produced hydrochar, which we are still looking at in the lab at Edmonton. So with this, we're looking at the future direction where we can take this to, because initially how we started was to look at if we can give seedlings a launch box and take them to the field so we can have higher quality stocks that can do better for reclamation purposes. However, when we looked at circular economy, so we're looking at how far can we take this to. So first of all, we're thinking of improving the hydrochar production processes. More or less, we need to understand if different hydrochar types will have different effects on seedling growth. Because with this hydrochar, you use a particular biomass. Sometimes you can use a biomass or a feedstock that has more nitrogen than the others. So we're trying to figure out if you have different feedstocks of hydrochar, whether we can start to see differences in those results I discussed earlier. Also, it would be important or it would be interesting to test this nutrient-loaded seedlings in field conditions over multiple years. So when we test this, we need to find out or fully demonstrate the impact of amendment, because having this in the greenhouse for 6 months does not represent what you can have in a natural field environment. So if we can have this in the field environment, you can start to even see differences. Ordinarily, we have not seen in the short period of time with the hydrochar in the greenhouse, we can start to see them in the field, vice versa, the alfalfa pellet. So a suggestion is to look at these quality seedling stocks in the field and see how far they can go. We can also look at other sources of organic amendments, especially from waste. Because when we're looking at organic amendments from waste, what we're going to do is we're diverting these materials from the landfill. So in as much as we're improving quality seedling stock and taking them to the field to achieve repollination, we're also promoting the circular economic approach to seedling production. Before I wrap up this presentation, I want to acknowledge the team that must be the hard work for this project. We have our second-year forest technology students that worked all day to do the harvesting and some of the measurements. And we have our technical staff that worked at Edmonton for preparation of this hydrochart, and those that worked at Fish River for the growth phase of this study. So I'll be open to answer any questions if we have time for that, or if we can have the questions of the general session. Thank you.